Okie doke. So let us round out navigation by talking about this question about if there are any other neural structures that are involved in navigation. And now we're going to move to a part of the brain that is referred to as entorhinal cortex. So entorhinal cortex is very close to hippocampus, but it is different. So if hippocampus is this sort of lighter gray matter section right here, kind of deeper in the middle of the brain, entorhinal cortex is kind of out here on the surface or the medial surface of the brain. So this is just a sort of schematic drawing to give you a real sense of what it looks like. These two highlighted parts here are entorhinal cortex, and this part here, that would be hippocampus, that would be hippocampus as well. Again, that's the corpus callosum. Okay, so within the entorhinal cortex is a group of cells that are not place cells, they're called grid cells, okay? Grid cells are a little tricky, they're a little weird. They're one of these things that I gotta be honest, I don't know how anyone would have ever predicted that they would have existed. I'm actually not entirely sure how people figured out that they existed, but the people who figured out that they existed, this married couple, Edward and Mary Britt Moser, they actually won the Nobel Prize. They shared the Nobel Prize for the discovery of grid cells. Okay, so a place cell will fire, for example, whenever an animal is on a particular location in space or in a particular place, okay? A grid cell will fire anytime an animal is on the vertex of a hypothetical grid. So for example, if you have an animal freely navigating around in this little circular area like this, you will find that the grid cells in the animal will fire every time, more or less, not always every time, but more or less, every time the animal is at this junction of this little triangular grid, okay? So now what I'm gonna do, just like before, I'm gonna show you an animal navigating around this box. This is the animal here, that's its tail. And now every time the grid cell fires, it's gonna be a little white dot. There's gonna be an experimenter over here every now and then throwing little pieces of food around just so the animal will navigate all over. So watch as this sort of grid builds up over time and the animal, as the animal is navigating around. So now we've just fast forwarded a bit to kind of speed it up. So you can kind of start to see the grid taking shape. And as the more the animal moves around, the more and more and more, you can start to see it even so, that they're kind of like these triangles. And that every time the animal gets on the vertex of one of these triangles, the grid is formed, okay? So this pattern of white dots is a pattern of firing for one neuron, okay? So if you are this neuron, basically you're always going to fire every time the animal is at the vertex of one of these grids, okay? Now, the grids are going to be very, very different from one another. It's not like it's gonna be all the neurons are gonna fire in the exact same way. So here, for example, are 4 different neurons, and you can see the firing pattern of 4 different scales, where over here, the grids are rather large. They're pretty big triangles. Whereas over here, the triangles are much smaller. It's a much finer grid. So it's operating at different scales of representation. Not only are they operating at different scales of representation, they're also interdigitated with one another. So here we've color-coded 3 grid cells, one in green, one in purple, one in red. And what you can do is you can kind of see that they're sort of intertwined, that it's like here's the triangle for a red one, more or less. And then here's the triangle a little off of it for a green one. Here's a triangle for a blue one, and so on and so forth. And so they're also kind of be interleaved with one another, or intertwined, as it were. Actually, and here's an even simpler way that you can see it. So it's actually have the green grid cell that's operating on this particular grid. Then you have in this case, now it's an orange grid cell operating on that grid, and the blue one, okay? And so basically what happens is that every time an animal or a creature enters a new environment, so like look at me in my box, let's imagine this is the environment, the animal will need a second to kind of get its bearing, and then the grid cell system will kind of line up to kind of scatter the new environment with all these little vertices, such that anytime the animal moves across the grid into this vertex, these cells will be firing accordingly, okay? So naturally, after people learned about this, they wondered, okay, are there grid cells in humans? So again, they did human electrophysiology, looking in human entorhinal cortex. And in this particular case, again, they were walking around these little virtual worlds, and they would have a task like, okay, in this virtual world, find the boat. And when they would do that, they would be on a little like hypothetical, bike, and they would find the boat and then they would go over to it. And yay, you've been teleported. Now find the hidden boat. So you got to kind of get your bearings based on where you were. They remember that the boat was in this corner, okay? Find the barrel, same environment. Where's the barrel? Where's the barrel? Where's the barrel? There's the barrel. See this big mountain in the background? So you kind of get your bearings of it. You've been teleported. Now find the hidden barrel. Get your bearings. Where's that mountain? There's that mountain. Come over around here until you find the barrel. Okay, so this is the virtual task that you're doing. And again, now while you're doing it, you've got these chronically implanted deep depth electrodes, except now they're not going into the hippocampus. Now they're going into entorhinal cortex. And here you can see the firing patterns on this hypothetical grid. So the color-coded jet color scheme in the background, that's the actual data. And then the experimenters put the X's on to kind of mark, where the firing patterns were. And so you can see, sure enough, as this patient is moving around, in this virtual environment, you can see the firing pattern mimic that of what we were seeing with the rat a couple of slides ago. So it seems actually as if in this sort of, medial temporal system, you have a combination, both in humans and in animals like rats, of both the hippocampus and the entorhinal cortex that are working together, where you have, if you're in this new environment, they're kind of doing complementary things, where the hippocampus will represent or will have these place cells that will represent where you are in one particular location in space, whereas the entorhinal cortex has these cells that are representing where you are in this hypothetical grid. So then the last question to think about is, are there other sorts of cells besides place cells and grid cells? In fact, there are two more other ones we can just talk about real quick. One of them are called head direction cells. And actually, you might wonder, what's a head direction cell? A head direction cell will fire depending on the direction in which your head is actually looking. So for example, if you are a rat and we're measuring a head direction cell, you'll see that cell 1 will fire when the animal moves its head to the left. Cell 4 will fire when its animal moves its head to the right. But it's not about body, it's about head direction. So everyone look down at me in the corner. Right now, my body is moving, but my head more or less is staying in place. So in that case, a cell would be firing in the same way, even as my body changes. And so, for example, if you're looking at a cell that's a forward-facing cell, like we'll call it cell 3 in blue, when the animal is looking forward, that cell will fire a lot. When the animal looks a little bit to the left, that cell will not fire as much in both directions. So that's some hypothetical data. Here's what some real data looks like. So when you're looking forward, you can see that this head direction cell is also firing maximally when you're looking forward, but as you start to look further and further to the left or further and further to the right, it will not fire nearly as much. I wanted to ask you all, if this looks familiar to you about anything else we've learned about, okay? Does this sort of plot maybe remind you anything? Because we've seen plots like this before. We've seen plots like this, for example, when we were talking about frequency selectivity in audition, where if you have an auditory neuron that is maximally responsive to, say, 1500 or 1700 hertz, as you go like lower frequency or higher frequency, the firing rate will drop off. We saw something similar to this in vision with orientation selectivity, where if you have a ideal orientation that a cell in V1 likes, it will not fire as much as you rotate it to the left or rotate it to the right. And you can see a similar thing even so in a head direction cell, where as the animal moves its head left or its head to the right, it will fall off accordingly. So you can see a sort of kind of generalizable principle emerging across domains of the brain, in audition, in vision, and now in this case in navigation, where neurons will have very peculiar and precise and idiosyncratic selectivities. And as you kind of break away from that selectivity, the neuron will kind of fall off in its firing rate accordingly. So that's a head direction cell. Like I wanted to really emphasize, it's not a body direction cell. That's something people often confuse. And again, you can think of this as being a part of this navigation system, where the hippocampus has the place cells, the entorhinal cortex has the grid cells. The head direction cells are in a part right nearby called the subiculum. And in this particular plot right here, I just want you to take a look at it. The reason that you can see that it's firing here, here, and here is that it's trying to say that this is the route that the animal was moving. And these are 3 spots where the animal happened to be looking up and to the left. And so it was firing in that case. And then the last one of these is what is called a border cell. And what a border cell is it won't respond when an animal is in a particular place or facing a particular direction or on a particular part of a grid. It's when an animal is up against a border of any form will it fire. So in this case, if you have, say, a wall here, so the animal can walk to the left or to the right of the wall, it will fire whenever the animal is at the lower part of the box or when the animal is up against this lower side of the wall. It will not fire as much when the animal is on the other side of the wall, though. So it's only this one particular border. It's like the southern border or something like that. And so you can put the animal in different types of shaped boxes, and it will still fire as a function of the same sort of southbound border. And this structure, I couldn't find a nice drawing like this, but these border cells, like the head direction cells, are also in this thing that is referred to as the subiculum. The one thing I want to say real quick is that as far as I know, I could be wrong. If someone finds it, please let me know. I'll add it to my lectures for next year. Head direction cells and border cells have not yet been found in humans, even though they have been found in rats. So that's why I'm not showing you any human ECOG data that has these. So then overall, we've seen in these sort of subcortical structures, these place cells, grid cells, head direction cells, and border cells. And something that I hope that you've gotten from this, alongside everything we were talking about with the PPA, RSC, and even to a certain extent OPA, which you'll again read about, is that navigation in the brain is broken down into many unique subsystems. It's not just a one sort of centralized navigation region in your brain that does all things. You have all these different unique subsystems all across the brain that are each working independently to compute particular aspects of navigation. Because if you remember, one of the very first things that I talked about, I think it was at the beginning of lecture 2, is I was saying, like, imagine you need to close your eyes and point to something like the front door of the science center, the gymnasium, the football stadium. In order to do that, you need to know a map of the world. You need to know where you are in it. You need to have a sense of the location of the target. You need to know your heading. You need to understand all these things. And so it actually turns out that to do all these different bits, you have a wide variety of circuitry in your brain across different parts of your cortex that are involved in doing these different subsystems. Okay, so we will stop there on navigation. The next section that we're going to talk about is going to now transition to memory. And that's when we're going to talk about different subsystems that are involved in long-term memory, short-term memory, and as I'm sure I already mentioned, as I'm sure a lot of you have already put together, in the same way that the hippocampus plays a big role in navigation, it plays a much larger role in memory. So stay tuned, and we will transition to the next part of it, which will be memory.